So as we continue to look at the internal workings of the eukaryotic cell, downstream in the endomembrane system from the ER is the Golgi apparatus, or one potential destination. It's the major place that most of the proteins that are being processed in the ER head to next. And the Golgi apparatus then is this kind of greenish blue pack or stack of pancakes that we see depicted here in the eukaryotic cell. So again, remember, the Golgi is part of a larger endomembrane system that consists of the nuclear envelope, the endoplasmic reticulum. Remember, there's rough and smooth. Now the Golgi apparatus. And downstream from the Golgi apparatus, one of the functions of the Golgi is to send proteins either to lysosomes, to vacuoles, or to the plasma membrane. So again, we can see that here is in the Golgi apparatus is where these arrows diverge. Right? So they can go on different paths from the Golgi. So the mechanism of the Golgi is going to allow us to send a protein to where it needs to go. So it's going to function to further modify the products of the ER. Proteins that are glycosylated get more, we think about it as like polishing the sugars that have been attached to the proteins in the ER are now modified further in the Golgi. It's going to manufacture certain macromolecules, and again, it's going to sort and package materials to transport vesicles. And so when we look at the Golgi in context of the cell, we can see that it has a cis side and a trans side. The cis side being the side closest to the ER, and the trans face, the trans side, being further from the ER. So as proteins enter the Golgi, they're going to proceed through the different chambers of the Golgi, each modifying them differently until ultimately they're transported and shipped out of the Golgi to one of their final destinations.